In the heart of every cell lies a remarkable molecule called DNA. This molecule holds the blueprint of life. Imagine a vast library filled with ancient scrolls. These scrolls contain the instructions for building and maintaining a living organism. DNA is like the language of these scrolls, a code written with 4 chemical letters, A, T, C, and G. These letters pair up in a specific way, A with T and C with G, forming the rungs of a twisted ladder. This ladder is the iconic double helix structure of DNA. The sequence of these letters along the DNA strands is crucial. It dictates the production of proteins, the workhorses of the cell. Proteins carry out a wide range of functions, from transporting molecules to catalyzing chemical reactions. They are essential for the structure, function, and regulation of the body's tissues and organs. The journey from DNA to protein, however, is a fascinating tale of cellular communication and molecular machinery. This process, known as gene expression, ensures that the right proteins are made at the right time and in the right amounts. It is a fundamental process that underpins all of life as we know it. Through the marvels of molecular biology, the instructions encoded in DNA are faithfully translated into the proteins that shape and sustain us. The first step in gene expression is transcription. Imagine needing a specific recipe from a cookbook. You wouldn't want to take the entire book to the kitchen. Instead, you'd copy down the recipe. This is analogous to transcription. DNA resides within the cell's nucleus, a protective compartment. However, protein synthesis occurs in the cytoplasm, the bustling hub outside the nucleus. To relay the genetic instructions, DNA relies on a messenger molecule called messenger RNA, or mRNA. During transcription, a special enzyme called RNA polymerase binds to DNA at the start of a gene. The DNA double helix unwinds, exposing the sequence of bases. RNA polymerase then uses one strand of DNA as a template to synthesize a complementary mRNA molecule. As RNA polymerase moves along the DNA, it adds RNA nucleotides one by one, following the base pairing rules. However, there's a slight difference in the RNA alphabet. Instead of thymine, T, RNA uses uracil, U. So, an A in DNA pairs with a U in mRNA, while the other pairings remain the same, C with G and G with C. Once the mRNA molecule is complete, it detaches from the DNA, and the DNA zips back into its double helix form. This newly synthesized mRNA molecule carries a transcribed copy of the gene's instructions, ready to be translated into a protein. The mRNA molecule acts as a messenger, carrying the genetic code from the DNA in the nucleus to the ribosomes in the cytoplasm. But how is this code read and understood? This is where codons come into play. Imagine the mRNA molecule as a long string of beads, with each bead representing a nucleotide. These nucleotides are read in groups of 3, and each group of 3 nucleotides forms a codon. Each codon specifies a particular amino acid, the building blocks of proteins. The sequence of codons in mRNA determines the sequence of amino acids in the protein. This relationship between codons and amino acids is known as the genetic code. The genetic code is like a universal language spoken by all living organisms. With 4 different nucleotides, A, U, C, and G, there are 64 possible codons. Out of these 64 codons, 61 codons code for the 20 different amino acids found in proteins. The remaining 3 codons, UAA, UAG, and UGA, are stop codons. They signal the end of the protein coding sequence. Interestingly, the genetic code is degenerate, meaning that multiple codons can code for the same amino acid. For instance, the amino acid leucine is encoded by 6 different codons. This redundancy in the genetic code provides a level of protection against mutations, as changes in the third position of a codon often do not affect the amino acid that is specified. Now that we have the mRNA molecule carrying the genetic code, let's explore where the actual protein synthesis takes place. The ribosomes. Ribosomes are complex molecular machines found in the cytoplasm of all living cells. They are the protein factories of the cell, responsible for translating the genetic code from mRNA into proteins. Ribosomes are composed of 2 subunits, a large subunit and a small subunit. These subunits come together like 2 halves of a clamshell, sandwiching the mRNA molecule between them. The ribosome then moves along the mRNA one codon at a time, reading the sequence of codons and assembling the corresponding amino acids into a growing polypeptide chain. the ribosome has three binding sites for tRNA molecules: the A site, the P site, and the E site. The A site is where the incoming tRNA molecule carrying the next amino acid to be added to the polypeptide chain binds. The P site is where the tRNA molecule carrying the growing polypeptide chain is located, and the E site is where the tRNA molecule that has donated its amino acid exits the ribosome. The process of translation is highly coordinated, involving a complex interplay between mRNA, tRNA, and the ribosome. Each step is carefully regulated to ensure that the genetic code is accurately translated into a functional protein. Section 5. The interpreters, tRNA and anti-codons. In the process of translation, another key player enters the scene: transfer RNA or tRNA. These molecules act as interpreters, translating the language of nucleotides into the language of amino acids. Each tRNA molecule has a unique three-dimensional structure that resembles a clover leaf. At one end of the tRNA molecule is an anti-codon, a sequence of three nucleotides that is complementary to a specific codon on the mRNA molecule. At the other end of the tRNA molecule is an attachment site for a specific amino acid. For example, the tRNA molecule with the anticodon UAC carries the amino acid methionine. During translation, the tRNA molecule carrying the appropriate anticodon binds to the codon on the mRNA molecule. This ensures that the correct amino acid is added to the growing polypeptide chain. The tRNA molecules are constantly being recycled. Once a tRNA molecule has donated its amino acid, it is released from the ribosome and can be reloaded with another amino acid by a specific enzyme. The interplay between codons, anticodons, and tRNA molecules ensures the accurate translation of the genetic code. It's a remarkable example of molecular recognition and precision engineering within the cell. Section 6. From code to creation: building proteins. With all the pieces in place, mRNA, ribosomes, and tRNA, the process of building proteins can begin. Remember that the ribosome moves along the mRNA molecule like a train on a track, reading the codons one by one. As the ribosome encounters each codon, a tRNA molecule with the matching anticodon pairs up with it. This tRNA carries its designated amino acid. Now the amino acid that was attached to the previous tRNA gets linked to the newly arrived one. This forms a peptide bond, a strong chemical link between amino acids. Imagine it like adding beads to a string, with the ribosome as the guiding hand. This process repeats codon by codon, extending the chain of amino acids. Eventually, the ribosome encounters a stop codon on the mRNA. These special codons, UAA, UH, UGA, don't have matching tRNAs. Instead, they signal the end of the protein building instructions. Upon reaching a stop codon, the ribosome releases the completed polypeptide chain. This chain then folds into a specific three-dimensional shape, determined by its amino acid sequence. This final folded structure is what gives the protein its unique function. Section 7. When codes change: mutations. The genetic code, while remarkably precise, is not immune to errors. Changes in the DNA sequence, known as mutations, can occur spontaneously or due to environmental factors like radiation. These mutations can have a variety of effects on protein synthesis. Some mutations are silent. They might change a single base in the DNA, but the new codon still codes for the same amino acid. Others, called missense mutations, are more significant. These alter a codon, leading to a different amino acid being incorporated into the protein. The consequences can range from minor to severe, depending on the protein and the specific amino acid change. For instance, sickle cell anemia arises from a single missense mutation in the gene for hemoglobin. Then there are nonsense mutations, which are often detrimental. These mutations change a codon that coded for an amino acid into a stop codon. As a result, protein synthesis terminates prematurely, leading to a truncated, often non-functional protein. Finally, insertions or deletions of nucleotides can cause frameshift mutations. These mutations disrupt the reading frame of codons, leading to a completely different amino acid sequence downstream of the mutation. Frameshift mutations usually have severe consequences, as they drastically alter the protein's structure and function. Section 8. The significance of faithful translation. The accurate translation of genetic information from DNA to RNA to protein is essential for life. Every protein in our bodies, from the keratin in our hair to the insulin regulating our blood sugar, is a product of this intricate process. Errors in transcription or translation can lead to a wide range of diseases, highlighting the importance of fidelity in these processes. Consider the complexity of a developing embryo. The precise timing and execution of protein synthesis are crucial for the formation of tissues and organs. Even minor errors can have cascading effects leading to developmental abnormalities. The study of mutations and their impact on protein synthesis has been instrumental in understanding the molecular basis of many diseases and in developing potential treatments. Section 9. The future of genetic exploration. As we delve deeper into the intricacies of the genetic code, we unlock new possibilities in medicine, biotechnology, and beyond. Gene editing technologies like CRISPR-Cas9 hold immense promise for correcting genetic defects and treating genetic diseases. The ability to manipulate the genetic code raises profound ethical questions that we must carefully consider as we venture further into this uncharted territory. Understanding the mechanisms of transcription and translation is crucial for developing new antibiotics that target bacterial protein synthesis. Furthermore, exploring the genetic code of diverse organisms can provide insights into the evolutionary history of life on Earth. By unraveling the secrets of the genetic code, we gain a deeper appreciation for the elegance and complexity of life itself. The journey from DNA to protein, a testament to the power of molecular biology, serves as a constant reminder of the interconnectedness of all living things.